Hi, Miss Nikki here. Welcome to Chapter 10. We finally get to talk about muscles. Yay! Part 1 here, it's kind of basic. We're going to do some defining and we're going to do a little bit of overview before we start talking about actual muscle contraction. Here we have a list of the functions of skeletal muscles. We're going to kind of focus on skeletal muscles, and then at the end of the chapter we'll talk about how cardiac and smooth are a little bit different. But the functions include 1, body movement. We've previously talked about bones. We know that bones act as the levers. We know that there are muscles attached to the bones, and those produce the effort in order for us to lift the load. We can also make facial expressions, speaking, breathing, swallowing. All of these body movements are one of the functions of skeletal muscles. 2, the maintenance of posture. We talked a lot in arthrology about stabilizing joints. So specifically, we talked about tendons that actually help stabilize the joint by surrounding the outside or attaching directly to the joint capsule. 3, protection and support. We're going to see muscles in areas that are covering internal organs, holding them in place. Probably the best way to think about that are those abdominal wall muscles. We'll talk about the fact that we have 3 sets of abdominal wall muscles. We don't have any bones on the anterior portion of our abdominal cavity. So this is offering some sort of protection and support. 4, we do have sphincters. So we have circular muscles that control the passage of material from inside the body to outside the body. So these sphincters are controlled by skeletal muscle. We have some sphincters that are controlled by smooth muscle. Then we have heat production. So skeletal muscles, in order to contract, are generating heat. And that heat is what helps you maintain your body temperature. Here we have a list of characteristics of skeletal muscle. When I think of characteristics, I think of kind of special abilities, right? What makes skeletal muscle special? How is it able to do its job? Remember, previous slide we talked about function, so try not to get function and these special characteristics mixed up or confused. So 1, skeletal muscle can respond to a stimulus. So if I have a neuron, and that neuron sends a message, the muscle can hear the message. So the neuron sends a message and the muscle can receive the message. 2, conductivity. If I send the message, that message can travel down the cell membrane of the muscle. So we have the neuron sending a message. Now that message can travel internally inside the muscle. 3, contractility. Once we have the message internally, we can cause proteins or fibers within the muscle to contract. So we can shorten the muscle. That's contractility. And when we shorten that muscle, we are creating tension. And that tension can be used as effort to lift a load. Hopefully this is all kind of coming together since we talked about this previously in bones. The last 2 are more just kind of inherent in the muscle overall, right? Extensibility and elasticity. So what's great about muscle is I can stretch the muscle and then I can return it to its original length. This won't make a lot of sense until we talk about the separate phases. But if you have a muscle that's this long, I can stretch it out a little bit, and then I can contract it a little bit, and it will go back to its original resting length. Something to think about is that a skeletal muscle is an organ. And this should make sense to us because we mentioned those 11 organ systems all the way back in Chapter 1. And muscle was one of them. So when I say organ to you, hopefully we know that this is different types of tissues working together. And remember, if we have tissues, we have to have cells. And if we have cells, we have to have atoms. And if we have atoms, we have to have chemicals. You remember that complexity of life? Okay. So there's going to be several different types of tissue. There's going to be muscle fiber cells. So they say muscle fibers, but they're actually cells, and we'll look at that in a second. We have connective tissue, we have blood vessels, and we have nerves. Do we remember that skeletal muscle is vascular? And we said it's innervated. Muscle fibers are bundled together, and we just give it a name. It's called a fascicle. The whole muscle contains many fascicles, and then inside of a fascicle there are muscle fibers. So this is something that kind of trips people up a little bit, and you might hear me call it a muscle fiber cell, or I might abbreviate it this way. I want to make sure that you realize that a muscle fiber is also just an elongated muscle cell. So they use these two words a lot. It gets confusing. I will try to say muscle fiber cell every time. We do have a lot of components. We have a wrapping system. So we have epimysium, which is connective tissue that wraps around the entire muscle. We have paramysium. This is dense, irregular connective tissue that's wrapped around a fascicle. And then we have endomysium, and this is connective tissue that's wrapped around individual fibers. And the image coming up next will probably help a little bit with this, but remember, epi is upon, peri means around, endo means inside. And anytime you see myo or mesium, you should be thinking muscle. So here we have skeletal muscle. So this entire structure here is one skeletal muscle. The skeletal muscle is wrapped in epimysium. Then we have fascicles, and each of these that I'm circling, these are bundles, and these bundles are wrapped in paramysium. Then inside of a fascicle, we have individually wrapped muscle fiber cells. So here's a muscle fiber cell, 1, 2, 3, 4, 5, and they're wrapped in endomysium. But that's not everything we need to know. We have to pull out, this is a muscle fiber cell. We have to pull this out, and we have to talk about what's inside here. So we're going to go even deeper into the muscle. We know that the attachment of muscle to bone happens via connective tissue called tendons, specifically dense regular connective tissue. So we can form a cord-like structure. We can also form a flattened sheet. So we'll see this aponeurosis, these flat sheets, when we move through the next chapter and we look at specific muscles. There is also deep fascia and superficial fascia. So on the last image, we saw epimysium, right? So epimysium is the connective tissue that surrounds the entire muscle. Then on top of the epimysium, so superficial to the epimysium, we have deep fascia. This is kind of just a way to organize muscles, right? To bind muscles with similar functions. And then on top of the deep fascia, we have another layer, which is called superficial fascia. So if I was going to kind of start on the outside, start superficially and work my way in, I would have superficial fascia, deep fascia, epimysium, then I would have my whole muscle, right? Then if I went further inside the muscle, what would be next? Paramysium. And what does paramysium wrap around? Fascicles. And then in fascicles, we have endomysium wrapping connective tissue around individual muscle fiber cells. I mentioned these terms previously. Do we remember skeletal muscle is vascularized? So that means it has many blood vessels. It is also innervated, meaning it has lots of neurons. Well, we just said in the previous slide that neurons are going to tell muscles what to do. If neurons from our brain are going to tell our muscles what to do we have to be able to get the neuron message to the muscle? Yes. So skeletal muscles have lots of innervation or lots of neurons. Skeletal muscle is vascularized because it needs to generate lots of ATP in order to do work. So in order for a muscle to contract, we need ATP. Remember, this is the molecule that we store energy in, that we use to do work within the body. But what do you need to go back to make ATP? We need oxygen, we need glucose, so that we can carry out cellular respiration and make ATP. Okay, here's the organization of a skeletal muscle fiber cell. So remember, we started with the muscle, then we have bundles, which are fascicles, and then we have muscle fiber cell. Well, we've expanded that. So this entire structure is a muscle fiber cell. And now we need to look internally. Inside every muscle fiber cell, you're going to have myofibrils, which are bundles of myofilaments, and these are proteins. If this is a cell, what does it have to have? All cells have a cell membrane or a plasma membrane. They all have a nucleus, and they all have cytoplasm, right? Those are at least the minimum qualifications. So here we have a plasma membrane or cell membrane. We specifically name it the sarcolemma. We have many nuclei. So here's another one over here. Does it make sense that we have lots of mitochondria inside a muscle fiber cell? Hopefully, from what we just talked about, we know this is the site of ATP generation from cellular respiration, specifically aerobic cellular respiration. So inside of a muscle fiber cell, we have nuclei, we have mitochondria, we have myofibrils, which are bundles of myofilaments, which are proteins. We also have something called sarcoplasmic reticulum. That should sound familiar. Endoplasmic reticulum, except for this time it's sarco. So instead of saying the endoplasmic reticulum inside a skeletal muscle fiber cell, I can just say sarcoplasmic reticulum and you immediately know what I'm talking about. We also have this structure called a triad. A triad consists of one T-tubule here in green, and then the terminal cisternae, which are in purple. Cisternae is kind of like a storage tank. So let's go to the next slide. We'll talk about all of these terms, and then we'll come back and look at them again. So a muscle fiber cell has some organelles and contractile proteins. It has multiple nuclei. Individual cells are multinucleated. So how did we get this one cell? We're used to a cell that looks this size, right? How did we get this one really long muscle fiber cell? Well, when this one cell decided to copy itself, it didn't split. If you remember cytokinesis, it decided, okay, we're not going to do that. It was programmed to not split. And now we have these really elongated muscle fiber cells filled with proteins, myofilaments, and lots of nuclei. Sarcolemma is the same thing as a plasma membrane or cell membrane. We have T-tubules that extend deep into the cell. We have also voltage-sensitive calcium channels. So what should you be thinking about? We just said in a previous slide, the neuron sends a message to the muscle, and then the muscle takes that message to the interior. So we're trying to take a neuron message, turn it into a muscle message, and then send that message internally. So here we have the muscle fiber cell. On the outside it has a plasma membrane, sarcolemma. What would be on top of the sarcolemma? Endomysium. Inside a muscle fiber cell, we have bundles of myofilaments, which are proteins. We have a triad, which is one T-tubule and two terminal cisternae. And we have this sarcoplasmic reticulum spread all around these myofilament proteins. We then label specific sections of these myofilament proteins, and this is called a sarcomere. It's just a specific section, and we'll come back and talk about this again. Can you see how if we have a neuron message or stimuli trying to tell this muscle to contract, I want you to do something. I want you to contract and generate force and lift a load. So the neuron message comes into the skeletal muscle, but I have to get that message all the way to the inside, deep internally. The sarcomere is the very first section that's going to contract. So this sarcomere is going to contract, then this sarcomere is going to contract, and this is going to happen all along the myofilaments. So neuron sends a message to the muscle then sends the message deep, and then we will have contraction. Some more terminology here. A myofibril is a bundle of myofilaments. They're wrapped in sarcoplasmic reticulum. It's very similar to smooth ER or smooth endoplasmic reticulum. We have terminal cisternae. We have a triad, which includes both terminal cisternae and a T-tubule. The terminal cisternae is going to store calcium. We're also going to see some calcium pumps, and we're going to see some calcium channels. They're kind of setting the stage for muscle contraction phases. So now we're probably totally confused, but let's just think about it for a second. Here is our muscle fiber cell. Inside of a muscle fiber cell, we have myofibrils. Myofibrils contain bundles of myofilaments. Myofilaments are what? Proteins. Certain sections of the myofilaments are the ones that are actually going to contract. So you're going to see this section get smaller. Then you're going to see this entire myofibril get smaller. Then what's going to happen to the muscle fiber cell? It's going to get smaller. What's going to happen to the fascicle? Smaller. Entire skeletal muscle contracts. Here are the T-tubules. So the T-tubules are here in green. The terminal cisternae flank both sides, and the terminal cisternae store calcium. Now we get even more technical. Don't get scared. This is our sarcolemma. So what are we talking about? If I have a sarcolemma, I have to be talking about a muscle fiber cell. They want to show you that there are specific gates or channels that are going to open and close, that we're going to see ions shift from outside to inside. And that shift of ions is going to take the message to the interior of the muscle. So remember, our whole goal is to take a neuron message, turn that into a muscle message, take that muscle message deep or internal, and then eventually we're going to have contraction. Here's a look at a triad. A triad is one T-tubule, and then it is two terminal cisternae. Inside the terminal cisternae, we are storing calcium, and we'll learn why in just a second. Now we need to talk about myofilaments. We have thick myofilaments and thin myofilaments. Thick myofilaments have myosin protein, and thin myofilaments have G-actin and F-actin. In addition, they have these regulatory proteins called tropomyosin and troponin. Okay, now we have to go even deeper. So here we are at muscle fiber cell. Muscle fiber cell is made up of myofibril bundles. What's inside the myofibrils? We have myofilaments. There are two different kinds of myofilaments. There's the thick myofilament and there's the thin myofilament. Sometimes they drop the myo. The thick myofilament is made up of the protein myosin. The thin myofilament is made up of two proteins, G-actin and F-actin. Sometimes people will refer to the thick as golf clubs. They kind of have this long tail, and then they have this nice head structure. For thin myofilaments, some people say this is beads on a string. So you cannot see the F-actin, this is a fibrous protein, and G-actin is a globular protein. So think of like beads that are strung on a string. You can't see the string, but the string is still there. And then there are two regulatory proteins, troponin and tropomyosin. People refer to tropomyosin as the gate and troponin as the lock. So tropomyosin stops unwanted contraction. Remember how we said skeletal muscle is voluntary. You only contract a muscle when you want to. So the gate, or the tropomyosin, stops unwanted skeletal muscle contraction. Only when we tell it to contract will the muscle contract. And the tropomyosin is like a secondary mechanism to control unwanted action. And that is kind of like a lock. And you'll be excited to see that calcium is the key. Where did we store calcium? Terminal cisternae, right? So this is all going to come together, I promise. So let's review all of these images kind of together. So we're starting with the entire skeletal muscle. So we're here at superficial, and we're traveling deep. So we know skeletal muscles are made up of bundles called fascicles. Fascicles are made up of bundles called muscle fiber cells. Muscle fiber cells are made up of myofibrils. Myofibrils are made up of myofilaments, which specifically are thick myofilament and thin myofilament. The thick myofilament is made of myosin. The thin myofilament is made up of F and G actin, and it has the two regulatory proteins, tropomyosin and troponin. Remember, if we're trying to send a message, so we have a neuron here delivering a message. My brain says, wiggle your finger. That message goes to the muscle, and that message has to travel deep into the muscle, because what's actually contracting? Individual sarcomeres. So specific sections of the myofilaments are contracting at a time, and that is being spread over the entire muscle and having the muscle contract. So how do I get the neuron message from the outside? How do I get it deep internally? The message is going to spread across the sarcolemma. So here they're showing you the sarcolemma. The message is going to spread across the sarcolemma. It's going to cause channels to open. That's going to cause the message to travel down the T-tubule. As the message travels down the T-tubule, calcium is going to be released, and that calcium is going to bind the troponin and remove the lock. So we're basically describing how do we turn a neuron message into an internal deep muscle message. And then eventually we're going to talk about contraction. Sarcomeres are repeating units of a myofilament. Some people think of them as the smallest contractile unit. This is where contraction starts, and then it spreads across the entire muscle. On both ends, we have something called a Z disc. It's an anchor. We organize the sarcomere, and these aren't as important to me. So the I band, the A band, the H zone, the M line, if you want to review these next couple of slides, you can. I really want to jump to the figure and talk just about the sarcomere. Here's a muscle fiber cell. We've stripped away all of the other myofibrils, so you can see just one. So remember, muscle fiber cells are made up of myofibrils. Inside myofibrils, we have myofilaments. Specifically, we have thick myofilaments and we have thin myofilaments. A sarcomere has two borders. It has a Z disc and a Z disc. These are anchoring points for proteins. On the next slide, we'll see a little bit more. But I wanted you to see these are repeating. So there are several sarcomeres in one myofibril, and then there are several myofibrils. So when I say the sarcomere is what's actually contracting, several sarcomeres can contract at once. It's not just one sarcomere and that makes your entire muscle contract. You're going to have many sarcomeres contracting to generate enough tension to cause the entire muscle to contract. Now we have a stripped down version of a sarcomere. I wanted you to see one sarcomere. Remember, it's bordered by Z disc to Z disc. Z disc is the anchor. You can see the thin myofilament is attached to the Z disc. Then we have this green structure, which is an elastic protein. And that is attached to the thick. So this is the thick myofilament. So the thick and the thin myofilaments are attached to the Z disc. Z disc to Z disc is what's going to contract. So a sarcomere is the smallest contractile unit, and this is where we're going to see the initial contraction event. All of the structures we've been talking about are directly related to the contraction event. We also have to acknowledge that there are some associated structures. So mitochondria is one of those structures. So muscle fiber cells have to have tons of mitochondria because we have to do aerobic respiration, right? Aerobic respiration generates that 36 to 38 ATP molecules, and the ATP, that's the stored energy we're going to use to do work in the body. So we have to make sure that we have tons of mitochondria to carry out aerobic respiration. We also store oxygen and we store sugar. Remember, glycogen is stored glucose. So if you remember, what do we need for aerobic respiration? For aerobic respiration, we need oxygen and we need glucose. So would it make sense if the muscle needs to have a ready supply of ATP, would it make sense for them to have the two starting materials or the two reactants necessary to make ATP? It's kind of like, well, duh, yeah, and we want to store those. We use it a lot. We're going to talk later about this creatinine phosphate. Some people say creatine, but it can give us a quick phosphate group that can help us make ATP really quickly. So when we get to the energy section, we'll talk a little bit more about this, but realize that we kind of focus on all of the proteins involved with contraction, but there are a lot of associated structures that we have to have in order to have a quick, ready energy supply. We need to talk about innervation real quick because we know we're taking a neuron message and we're kind of translating it into a muscle message, right? So here we're showing the spinal cord and a spinal nerve, and you can see the neurons are reaching out and they're making contact or coming into very close association with muscle fiber cells. So this is called a neuromuscular junction, where the neuron is coming in contact with the muscle. So this is going to be the site where we first transfer this message. We're going to take the neuron message, we're going to turn it into a muscle message. And then what's the muscle going to do? The muscle's going to take that message internally. We have to get all the way to the inside, right? What do we have to get to? We have to get to sarcomere. The sarcomere is what's actually contracting first. So the sarcomere contracts, that spreads over the entire muscle fiber cell, and then we have the entire contraction of the muscle. We have small motor units and we have large motor units. So a small motor unit can talk to usually less than 5 muscle fiber cells. Large motor units can have thousands of muscle fiber cells that it's talking to. So this would be the difference. Let's say small motor unit would control eye blinking. precise control. You don't need tons of force. So sometimes I say low force. If that trips you up, just ignore it. But large motor units, I'm trying to generate a large amount of force, a large output. This would be like kicking a door down. If you're using your quads, right, to forcefully kick a door down, then you're engaging large motor units that are controlling thousands of muscle fibers and telling thousands of muscle fibers to contract all at one time to generate enough force to lift your leg and kick the door down. Here's a neuromuscular junction image. Again, here's a motor neuron, and this motor neuron is reaching out and talking to 3 muscle fiber cells. And the way that it does that is by a synaptic knob. Sometimes I think of this as a foot, right? The foot of the neuron. So this synaptic knob is going to sit in really close proximity to the sarcomere of the muscle fiber cell. Sometimes they call it a motor end plate. We know we have our bundles of myofibrils, and then what's inside the myofibrils? We have myofilaments. On the neuromuscular junction, we have that synaptic knob. It's the tip or the foot of the motor neuron. It has vesicles, small sacs, and they're filled with acetylcholine. It has calcium pumps in the plasma membrane, that's of the neuron. And there's a calcium channel in the membrane. Here's a close-up of the neuromuscular junction. Here's our synaptic knob. This is the foot of the neuron. It has vesicles that are filled with acetylcholine. This is a neurotransmitter, and that's what's going to send the message to the muscle. So again, I know I keep repeating myself, but what are we doing? We're taking a neuron message and we're turning it into a muscle message. What's doing that? The acetylcholine. So the transfer of acetylcholine is how we turn a neuron message into a muscle message. So we're going to learn more about this. I just kind of wanted to show you the neuromuscular junction and how the neuron here, the synaptic knob of the neuron, comes in close proximity to the muscle fiber cell. And here's my sarcomere, my phospholipid bilayer plasma membrane. There's also a structure called a motor end plate. We've seen this already, and it has acetylcholine receptors. There's also a structure called a synaptic cleft. This is kind of the space between them, between the neuron and the muscle. So again, same image. Here's our synaptic knob of our neuron. Here is the plasma membrane, so this has to be our muscle fiber cell. We have the vesicle filled with acetylcholine, and the vesicle is eventually going to deposit the acetylcholine outside of the neuron. This is the synaptic cleft. So all of this space is the synaptic cleft. So do we see how the acetylcholine is dropped into the synaptic cleft? Then the acetylcholine is going to come over here and bind to the acetylcholine receptor. The acetylcholine receptor is embedded in the sarcomere of the muscle fiber cell. So basically, how are we turning a neuron message into a muscle message? We're using the neurotransmitter acetylcholine. It's almost like the neuron comes to the muscle and says, hey, I have a message for you, and then it passes off the acetylcholine. Now the muscle is going to pick up the acetylcholine, and some actions are going to happen that we're going to learn about. These last 2 slides are kind of the last pieces of the puzzle before we're going to go through all the phases and walk through all the steps that have to take place for a muscle to contract. Hopefully you remember from a while ago we talked about resting membrane potential. So when a muscle is at rest, the inside is at about negative 90 millivolts. The outside is positive. And it's positive because there's lots of sodium on the outside of muscle fiber cells. What's on the inside? On the inside, there's lots of potassium and other really large negative molecules, phosphates. So the inside of a muscle fiber cell is negative. For this last slide, let's do an overview. In part 2, we will cover all of the phases. So I'll go into more detail and we'll kind of do a bullet point of what we need to know. But right now, what have we covered in this slide or in this part 1? We have a neuron message and we're turning it into a muscle message. How did we do that? How did we turn a neuron message into a muscle message? We took this acetylcholine and we transferred it from the neuron to the acetylcholine receptor on the muscle fiber cell. So think of the acetylcholine as kind of like the baton. I'm turning this over to you. We're doing a relay race. I'm giving you a message. I'm giving you something to take and run with. So the acetylcholine is going to bind to the acetylcholine receptor, and that's going to generate something called an action potential. That action potential, that message, we now have a muscle message, but where do we have to get the muscle message? The muscle message has to get deep, internal. Where does our contraction start? It starts at the sarcomere. So we've generated a muscle message, now we have to get it inside, deep to the muscle. So this action potential is going to travel through the T-tubule, and along the way, calcium gets released from the terminal cisternae. The calcium will come down here and bind the troponin and move the gate, which is the tropomyosin, and now the myosin heads can reach up and pull on the thin myofilament, and we can actually have contraction. Hopefully that made a little bit of sense. We will go through this line by line in part 2. I'll talk to you soon. Bye.